Imagine a massive, bustling metropolis that's operating at just absolute peak efficiency.
Right.
You know, you have factories churning out vital materials. You've got a highly regulated highway system with cargo trucks just zipping back and forth. You have demolition crews taking down old structures to make way for new ones, and a central command dictating the timing of every single event.
Yeah, typical city.
Exactly.
Okay.
But now imagine that this entire city is functioning perfectly without a single wall, fence, or building anywhere.
Oh, wow.
There are no secure perimeters. There are no locked doors. Everything is happening entirely out in the open in this shared fluid space. Yet it's all orchestrated with absolute life or death precision. I mean, that is the human oocyte. And that is the landscape we are doing a deep dive into today.
It really is a totally different way of looking at it.
Because, you know, we spend so much time in biology just fixating on borders. We love the lipid bilayer. We love the nuclear envelope keeping the DNA safe, or the double membranes of the mitochondria, or the stacked lipid folds of the Golgi apparatus.
Right. Membranes give us this comforting sense of compartmentalization. Everything has its own room.
Exactly. But when you are actually looking through the microscope at a fertilized human egg, some of the most critical events deciding the fate of that potential life are happening completely outside of those membranes.
Yeah, it forces a complete mental shift in how we understand cellular architecture. I mean, we are so conditioned to think that biological complexity requires physical isolation, that to do a specific job, a cell needs a dedicated membrane-bound room.
Right.
But the early embryo proves that is simply not the case. So today we are focusing on these molecular machines that assemble spontaneously in the cytoplasm, execute highly specialized catalytic or structural roles, and govern the development of gametes without a single phospholipid molecule holding them together. We are talking about the non-membrane-bound organelles.
And for those of you listening, especially the clinical embryologists prepping for the ESHRE exam, or really anyone who spends their days managing incubators and micromanipulators, the stakes here are just absolute. We aren't just talking about abstract cell biology.
No, not at all.
Think about the last time you looked through the scope at a day 1 zygote and saw a single pronucleus, or an embryo that looked gorgeous at the cleavage stage, but completely arrested on day 3. Your first instinct might be to blame, say, sperm DNA fragmentation or an inherent chromosomal aneuploidy.
Which is a fair assumption, but
But so often the failure is entirely mechanical. It is a failure of these open-air structures, the translation machinery, the cytoskeletal tracts, the protein degradation barrels. When these non-membrane-bound structures fail, the entire city collapses.
which is exactly why our goal today is to systematically decode these structures. We need to look at their internal mechanics, understand how they organize the cytoplasm, and most crucially, identify what it looks like on the lab bench when they break down.
Because the embryology exam loves to test the boundaries of normal function versus pathology.
Oh, absolutely. They love it. And there's no better place to start than the literal origin point of every protein in the cell.
Yeah.
The factory floor itself.
The ribosome, the
Yeah.
universal translator. Because no matter what happens later, whether we are talking about the enzymes that drive metabolism, the motor proteins that move cargo, or the tubulin that makes up the meiotic spindle, every single one of those proteins has to be physically assembled by a ribosome.
And the sheer scale of the operation is staggering. I mean, a single mammalian cell can contain up to 10 million ribosomes.
10 billion, that's insane.
It is. And they are massive, dense ribonuclear protein complexes. That means they are a hybrid structure, like this intricate puzzle assembled from dozens of individual ribosomal proteins and several long strands of ribosomal RNA, or rRNA.
You know, I remember learning about ribosomes early on and having a massive misconception about them.
What was that?
Well, because enzymes are almost always proteins, I just assumed the protein parts of the ribosome were doing the actual work of building the new amino acid chain. And I figured the RNA was just there as a sort of structural skeleton, you know, to hold it all together.
Right, that makes logical sense. But historically, that assumption was completely turned on its head.
It really was.
It was one of the most profound discoveries in molecular biology, actually. Researchers eventually crystallized the ribosome and mapped it down to the atomic level.
Yeah.
And what they found in the core, the peptidyltransferase center, which is the exact active site where the chemical bond is formed between two amino acids, contained absolutely no protein.
None at all.
None. It is entirely composed of ribosomal RNA. The RNA is the catalyst. The RNA is doing the chemistry.
Making it a ribozyme, not a traditional enzyme. It is just wild to me. It's like looking at a modern high-tech factory and realizing the central piece of machinery is a 3 billion-year-old stone tool that still works perfectly.
That's a great way to put it. It is a molecular fossil from the ancient RNA world, from way before DNA and proteins even existed.
It truly is. And because that RNA core is so fundamental to life, it's deeply conserved across evolution. Like a bacteria, a yeast cell, and a human oocyte all use the exact same fundamental mechanism to link amino acids.
Exactly.
Uh-huh.
However, the outer scaffolding, the overall size and architecture of the ribosome, did diverge over billions of years. And that divergence is an incredibly high-yield topic for anyone studying for the ESHRE exam, especially when we get into experimental pharmacology.
Okay, let's break down that architecture then. We classify ribosomes based on their sedimentation coefficient, which is measured in Svedberg units. Now, I have always found Svedberg units slightly infuriating.
Let me guess. The math.
Yes. The math doesn't seem to add up at all. You have a 40S subunit and a 60S subunit, and somehow they combine to make an 80S ribosome. 40 plus 60 does not equal 80.
Yeah, it is a really common point of confusion. The Svedberg unit, denoted by the letter S, is not a measure of mass. It's a measure of how fast a particle sediments or sinks to the bottom of a tube when subjected to extreme G-forces in an ultracentrifuge. The speed of that sedimentation depends on a combination of the particle's mass, its density, and its physical shape. Think about a skydiver. A skydiver with a closed parachute falls at a certain speed. If two skydivers hold onto each other, their mass doubles, but their aerodynamic drag also changes dramatically. They do not fall exactly twice as fast.
Oh, that makes perfect sense.
Right. So when the small and large ribosomal subunits physically lock together, their combined shape changes their drag coefficient in the centrifuge fluid. So a 40S and a 60S subunit combine to form a more compact 80S particle.
Got it. So in our human embryos, the primary cytosolic ribosome, the one floating out in the open cytoplasm, is this 80S eukaryotic form. And to pass these exams, you really need to know the components.
Definitely.
The small 40S subunit contains the 18S RNA strand. The large 60S subunit contains 3 strands, the 28S, the 5.8S, and the 5S. And they associate around the messenger RNA in a process that requires a very specific concentration of magnesium to remain stable.
That's a good detail to remember. But we also have to talk about the 70S form.
Right, the bacterial one.
Exactly. The 70S ribosome is the ancestral bacterial form. It's made of a 30S small subunit and a 50S large subunit. Now, you might ask why a clinical embryologist studying human reproduction needs to care about bacterial ribosomes at all.
Because it seems completely irrelevant at first glance.
Right. But the answer lies in endosymbiotic theory. Hundreds of millions of years ago, a primordial eukaryotic cell engulfed a free-living bacteria, which eventually evolved into our mitochondria. Because of that ancient heritage, human mitochondria still contain their own small circular DNA, and they still build their own 70S bacterial-style ribosomes to translate those specific genes.
So inside a human oocyte, which has upwards of 100,000 mitochondria, you actually have 2 entirely different populations of ribosomes operating simultaneously. You have the 80S factories in the cytoplasm and the 70S factories locked inside the mitochondria.
Exactly, and this is where the pharmacology comes in, which is huge for the exam.
Right, because their structures are different, their vulnerabilities are different.
Yes. Certain antibiotics, like tetracycline, chloramphenicol, and streptomycin, were developed specifically to wedge themselves into the 70S bacterial ribosome. They paralyze it. They stop bacteria from reproducing in an infection. Crucially, they do not bind to the human 80S cytosolic ribosome.
Which is why we can take them without all our cells dying.
Precisely. On the flip side, we have toxins like cyclohexamide, which specifically target and paralyze the 80S eukaryotic ribosome without affecting the 70S version.
So if you are setting up an experiment in the lab and you want to know if a specific protein is being made by the mitochondria or if it's being made by the general cytoplasm, you can use these drugs to selectively mute one factory while leaving the other running. It is an incredibly powerful tool for unraveling early developmental pathways.
It really is.
But let's shift to a massive logistical problem. If all the millions of cytosolic ribosomes in an oocyte are structurally identical 80S particles, how does the cell sort the output? I mean, a cell needs some proteins to stay floating in the cytoplasm, like the tubulin for the spindle, but it needs other proteins to be embedded in a membrane or secreted entirely outside the cell, like the zona pellucida proteins.
Right. How does it know?
Yeah, how does a blind RNA machine know where its product is supposed to go?
It is a brilliant mechanism, honestly.
Right.
The ribosome itself is completely agnostic. It doesn't care. It just binds to an mRNA transcript and starts building the protein, amino acid by amino acid. The routing code is actually hidden within the first few amino acids of the protein being built.
The signal peptide.
Yes. If a ribosome is translating an mRNA that codes for a cytosolic protein, say cyclin B, which we will discuss a lot later, there's no special signal. The ribosome simply finishes the job, releases the protein into the cytoplasm, and floats away to find another mRNA.
Okay, simple enough.
But if the mRNA codes for a secreted protein or a membrane receptor, the very first sequence of amino acids to emerge from the ribosome is a highly hydrophobic signal peptide.
So this hydrophobic chain pops out of the ribosome, and because the cytoplasm is an aqueous, water-based environment, that hydrophobic chain is essentially screaming for cover. It acts like a molecular siren.
That's a perfect analogy. And that siren is immediately answered by the signal recognition particle, or SRP. The SRP is a complex that patrols the cytoplasm. The moment it detects that hydrophobic signal peptide, it clamps down on it. Crucially, it also binds to the ribosome itself and temporarily freezes translation. It pauses the factory.
It stops production so the protein doesn't fold up incorrectly in the wrong environment.
Exactly. You don't want a hydrophobic protein misfolding in the cytosol. The SRP then physically drags the entire paused complex, the ribosome, the mRNA, and the partially built protein, over to the endoplasmic reticulum. It docks with an SRP receptor on the surface of the ER.
And then what?
Once securely anchored, the SRP releases its grip, translation resumes, and the newly growing protein chain is threaded directly through a pore into the protected interior space of the ER.
That is how we get the rough ER. I remember always wondering why it was rough. The studs on the rough ER are just normal cytosolic ribosomes that happen to be translating a secreted protein at that exact moment. They aren't permanently fixed there.
Right, they come and go as needed.
And in the context of an oocyte growing in a follicle, those membrane-bound ribosomes are working overtime. They are synthesizing all the zona pellucida glycoproteins, ZP1, ZP2, ZP3, ZP4, threading them into the ER so they can be packaged into vesicles and secreted to build the shell around the egg.
They're also building the contents of the cortical granules, which we know are essential for preventing polyspermy.
Right. Meanwhile, the free-floating cytosolic ribosomes are churning out the massive quantities of structural proteins and cell cycle regulators required for the mechanics of cell division. But to truly grasp the clinical vulnerability of the embryo, we have to look at the timeline of when these ribosomes are actually built.
Because ribosome biogenesis is arguably the most energetically expensive thing a cell ever does.
It happens in the nucleolus, right? Inside the nucleus.
Yes. RNA polymerase I transcribes a massive 45S precursor strand of rRNA, which is then heavily modified and chopped down into the smaller 28S, 18S, and 5.8S pieces we mentioned earlier. This process requires a tremendous amount of resources.
It's a huge undertaking.
If you look at a growing oocyte under high magnification, its nucleolus is enormous. It looks like a massive, dense sphere inside the germinal vesicle. It is working in absolute overdrive.
But why the frenzy? I mean, why does an oocyte need to build so many millions of ribosomes before it even ovulates?
Because it is preparing for a period of total transcriptional silence. The oocyte is employing an extreme survival strategy known as the maternal stockpile. It spends months, potentially years, quietly hoarding ribosomes, free ribosomal subunits, and thousands of different messenger RNA transcripts.
Let's track that timeline for the listeners, because this is where the clinical relevance spikes. The moment the oocyte receives the LH surge and undergoes GVBD germinal vesicle breakdown, when the nuclear envelope dissolves to resume meiosis, transcription essentially stops.
It grinds to a halt.
Right. The DNA condenses into chromosomes, so you can no longer read the genes to make new RNA.
It stops at GVBD. It remains stopped through the extrusion of the first polar body. It remains stopped during the M2 arrest. It remains stopped during fertilization, during pronuclear formation, during the first cleavage division, the second division.
All the way up to embryonic genome activation, or EGA, which in humans does not fully kick in until the 4 to 8 cell stage on day 3.
That is a staggering amount of time for a cell to live, move, divide, and function without being able to access its own DNA blueprints. For those first 3 days of life in the incubator, the embryo is entirely 100% dependent on the maternal stockpile.
It has no other option.
None. It must rely on the ribosomes and the mRNA the oocyte hoarded during folliculogenesis. If the embryo needs a specific protein to execute the first cleavage division, it cannot transcribe the gene for it. It has to pull an mRNA out of the stockpile and hand it to a pre-existing maternal ribosome.
Think about the quality control implications of that in the lab. If that maternal stockpile of ribosomes is damaged, there is no backup system. You cannot just build more.
There's absolutely no backup. And this is exactly where suboptimal conditions in the IVF lab exert their insidious effects. If your culture media degrades, or if the oxygen tension in the incubator is too high, it generates reactive oxygen species.
And those ROS do what?
Oxidative stress physically damages RNA. It introduces lesions into those stored maternal mRNAs, and it damages the ribosomal RNA in the catalytic core of the ribosomes themselves.
Wow. So you have a patient with beautifully mature oocytes, successful fertilization. You get two distinct pronuclei. The zygote cleaves to two cells, and then it just stops. Total developmental arrest on day 2 or day 3.
Cleavage arrest, it's heartbreaking.
Yeah.
And it is easy to assume the embryo had a catastrophic chromosomal aneuploidy. But very often, it arrests simply because of translational insufficiency.
The molecular factories just broke down.
Yeah, the damaged ribosomes physically cannot synthesize the proteins required to run the next cell cycle. The embryo literally runs out of the machinery required to build the next phase of its own life.
It is a really sobering reminder of how fragile that initial maternal endowment really is. Okay, so we have established the factories. The ribosomes are building the proteins. But a city cannot just be a pile of factories.
Right, you need infrastructure.
Exactly. It needs roads, it needs traffic control, it needs a scaffold to maintain its shape and move things around.
Yeah.
Which brings us to our next non-membrane-bound marvel, the centrosome and its core structural components, the centrioles.
If the ribosome is the factory, the centrosome is the central traffic control tower and the master architect of the cell's internal highway system. It's the primary microtubule-organising center, or MTOC, in animal cells.
So it's dictating all the movement.
Yes. Every vesicle of proteins moving from the ER to the Golgi, every chromosome being pulled apart during division, every pronucleus migrating to the center of a newly fertilized egg. It is all orchestrated by the microtubule tracks laid down by the centrosome.
The architecture of this structure is visually stunning when you look at electron micrographs. Let's build it from the ground up for the listeners. At the very core of the centrosome, you have a pair of centrioles, a mother and a daughter.
Yes, and they sit perfectly at right angles to each other, forming this L shape. Structurally, a centriole is a rigid, perfect cylinder, roughly 200 to 500 nanometers long. But the wall of that cylinder is not solid. It is built from 9 distinct triplets of microtubules arranged in a circle.
Let's clarify what a triplet means here, because that gets confusing.
Yeah.
The basic building block of a microtubule is a heterodimer of alpha and beta tubulin. Those dimers stack up like bricks to form a hollow tube called a protofilament.
Right.
A standard microtubule out in the cytoplasm requires 13 of those protofilaments arranged in a circle to form a complete tube.
Exactly. But in the centriole wall, it is a triplet. You have an inner A tubule, which is a complete 13 protofilament microtubule. Fused to the side of that is a partial B tubule, usually made of 10 protofilaments. And fused to the side of that is a partial C tubule, also 10 protofilaments.
Okay, so they're stacked onto each other.
Yes. 9 of these massive ABC triplets form the cylinder, anchored around a central structure that looks exactly like a cartwheel.
It is astonishingly complex, and we see this exact same 9-triplet arrangement across an incredibly wide range of species. It is deeply evolutionarily conserved. Now, you mentioned a mother and a daughter centriole. How do you tell them apart?
Well, the mother centriole is older, having been built in a previous cell cycle. Structurally, it is distinguished by specialized protein structures at one end called subdistal and distal appendages.
And what do those do?
These appendages are critical. They act as grappling hooks. If the cell needs to grow a primary cilium or flagellum, the mother centriole uses those appendages to physically dock against
Thank you sincerely for your patient listening and dedication to mastering clinical embryology. It's professionals like you who advance our field every day. If this content added value to your practice, a simple like helps it reach more embryologists and assisted reproductive technology specialists worldwide. Please subscribe and turn on notifications so you never miss updates on lab protocols, cryopreservation tips, and guideline-driven insights. Your support fuels more free, high-quality resources for our entire assisted reproduction community. Let's grow this channel together for better patient outcomes.